An eye tracker shows where a participant looks. EEG records the electrical activity of the brain. Physiological sensors measure signals such as electrodermal activity, cardiac activity, and respiration. Each measurement can be valuable on its own. The real challenge begins when we ask one question: what exactly was happening in the body? At the moment the participant looked at a specific part of the stimulus. If 3 devices record data against 3 independent clocks, even excellent signals may fail to form a reliable story. In this video, we will show how to plan a shared timeline for eye tracking, EEG, and physiology, what roles MP200, AcqKnowledge, LSL, TTL, and NDT play, and why an architecture designed for a screen-based study may not fit wearable eye tracking glasses. Imagine a simple experiment. A stimulus appears on the screen. 2 seconds later, the participant looks at an area of interest. Shortly afterwards, electrodermal activity changes, and an interesting pattern appears in the EEG recording. On a chart, this looks like one sequence. Technically, however, it may involve 4 different timelines: the stimulus software, the eye tracker, the EEG system, and the physiological recorder. Every computer and every device measures time a little differently. An initial difference is an offset. A changing difference over the course of a recording is drift. Network latency, buffering and processing time add further uncertainty. Synchronization therefore means more than pressing record at approximately the same moment. We need shared reference events, and we must know where each marker originates, how it is transmitted, and in which file it is stored. The more precise the timing question, the more important this path becomes. First, let us separate 2 things. Continuous data are streams of samples. Gaze coordinates, pupil size, EEG, ECG, EDA, or respiration. Events are markers that describe when a trial starts, a stimulus appears, a participant presses a button, or an experimental stage ends. A valid analysis needs both. A shared recording start does not tell us where a particular stimulus occurs in a long data file. Markers alone, without continuous signals, do not show the response between events. A good architecture therefore answers 3 questions. What is the time reference? How does each data stream reach the recording? How are experimental markers linked to every signal? Only then can we decide whether the study requires software integration, hardware TTL pulses, post-study import, or a combination of methods. BIOPAC MP200 is a central physiological data acquisition system. Depending on its configuration, it can record ECG, EMG, EEG, EDA, respiration, and other signals through the appropriate modules. The system supports up to 16 channels, sampling rates up to 100 kHz per channel, and an aggregate sampling throughput of up to 1.6 MHz. These figures do not mean that every study should run at the maximum rate. Parameters must match the signal and the research question. AcqKnowledge is the software layer. It is where channels are configured, recordings are monitored, events are added, and analysis is performed. In a multimodal laboratory, AcqKnowledge can become the environment where physiology is aligned with data from other devices. This does not happen automatically, simply because all devices are in the same room. It requires the correct licenses, interfaces, and a deliberately designed synchronization path. In practice, 4 mechanisms appear frequently, and they should not be confused with one another. The first is ACK100W-EYE. This license integrates compatible screen-based eye tracking systems with AcqKnowledge. It allows gaze data to be aligned with physiological recordings, and the current package also includes stimulus presentation functionality. It is a natural direction when a study is performed at a screen with a supported eye tracker. The second mechanism is ACK100-LSL. Lab streaming layer helps exchange streams and markers between research systems. In the current version of AcqKnowledge, this license receives LSL streams from other devices. It should not be described as a bidirectional bus that automatically publishes all AcqKnowledge data to the network. If an external application needs access to AcqKnowledge data, NDT or network data transfer is a separate solution. Direction matters. LSL can bring selected streams into AcqKnowledge, while NDT can expose AcqKnowledge data to an external system. The fourth mechanism is TTL. A short electrical pulse can mark stimulus onset or a participant response without relying on the usual operating system and network delays. TTL is not obsolete. In many setups, it remains a useful deterministic reference point. A strong design often combines hardware markers with richer event descriptions sent in software. Let us examine three common architectures. The first is a screen-based study. A computer presents the stimulus, a screen-based eye tracker measures gaze, and MP200 records physiology. If the devices are compatible with ACK100W-EYE, eye tracking and physiological data can meet inside AcqKnowledge. The stimulus application should send unambiguous markers for trial onset, presentation, and response. TTL is worth considering for timing critical events. The second architecture is a multimodal laboratory with an additional EEG system or another device that publishes LSL. AcqKnowledge can receive these streams through ACK100-LSL. Channel names, sampling rates, markers, and behavior after a connection loss should all be tested in advance. If another application needs to read live physiological data from AcqKnowledge, a separate path such as NDT is required. The fact that two systems support LSL does not remove the need to test the complete chain. The third architecture uses eye tracking glasses during natural movement, work with physical objects, or navigation through an environment. There may be no single screen and no fixed stimulus application. Data from the glasses and the physiological recorder may be collected separately and then aligned after the study using events, a shared signal, or a synchronization procedure. This workflow can be entirely valid, but it must be designed before the first participant. Otherwise, researchers may discover during analysis that there are no unambiguous alignment points. These examples demonstrate one principle. Do not choose an interface because its name sounds modern. Choose it according to data flow direction, required timing accuracy, and study design. Why do wearable glasses require separate consideration? In a screen-based study, the frame of reference is stable. The positions of the monitor, stimulus, and eye tracker are known. In a mobile study, the participant turns their head, changes distance, interacts with the environment, and a scene camera records a changing world. The challenge is therefore not limited to time. Gaze must also be mapped onto real-world objects. An event that has an unambiguous identifier on a screen may require video coding, environmental markers, or manual verification in a mobile recording. We should not promise automatic live integration without checking the particular model of glasses and software workflow. Before selecting a device, ask one question. Is the stimulus a controlled screen, or should the participant act freely in a real environment? The answer usually determines which class of system should be considered first. Before the first participant, perform 6 checks. First, document every device, computer, signal, and sampling rate. Second, draw the direction of data flow. Who publishes, who receives, and where is the final file created? Third, define an event dictionary. Block start, trial start, stimulus presentation, response, and trial end. Names should be unambiguous and consistent. Fourth, run a test with an easily recognizable event that is visible in several systems. Check the offset at the beginning and the end of a longer recording to detect drift. Fifth, simulate a fault, a temporary network loss, an application restart, or a missing stream. Confirm whether the system reports the problem and how data can be recovered. Sixth, complete a test export and analysis. Moving charts during recording are not enough. Markers, units, channels, and metadata must survive the entire workflow. Synchronization is only one dimension of quality. A high percentage of gaze samples does not automatically mean that data are ready for area of interest analysis. Samples may be numerous, yet systematically displaced, unstable in selected segments, or mapped poorly onto the scene. A quality report should therefore combine data completeness with accuracy validation, stability, sample loss control, and synchronization assessment. A multimodal study also requires consistent markers across systems. One attractive percentage cannot replace control of the complete measurement chain. The best time to find a failure is during the pilot, not after recruitment has ended. A few minutes spent testing the timeline can save weeks of unusable data collection. If you are planning a laboratory with eye tracking, EEG, and physiology, begin with the architecture, not a device list. Prepare 3 pieces of information: which signals you need to measure, which devices or applications you already use, and how stimuli will be presented. Send this list to Interlab. We will help you select the eye tracker, BIOPAC system, modules, licenses, and synchronization interfaces for the planned experiment. Links to the solutions and articles discussed in this video are available in the description.